Streff is a certified genetic counselor at Texas Children's Hospital and Baylor College of Medicine. She completed her master's degree in genetic counseling at the University of Texas Health Science Center in Houston. She sees patients in the outpatient genetic clinics and multidisciplinary craniofacial clinics at Texas Children's Hospital. She also works on genetic test utilization at Texas Children's Hospital and is a clinical supervisor at Baylor Genetics Counseling Program. Haley enjoys participating in outreach and education in the rare gene community, and she serves on a board of directors and multiple committees for various genetics and family support groups. Please welcome Haley Streff.
Okay, so I'm kind of doing this with Dr. Wangler. I'm going to go first and do more of like the 101 part. I would actually say, I think Dr. Chow did a pretty good job giving us some 101. So we're maybe going to move up and advance this year into like more 201, 301 type categories. And then I'm going to pass it off to Dr. Wangler, who's going to talk about kind of some of the questions we had about a little bit more about like therapeutics interventions. So we're going to cover more genetics. I think you're going to hear a lot of genetics. HADS is a condition caused by a change in our genes. So we're going to talk a lot about it. So again, this is kind of our path forward for the next however long it needs. The first thing I'm going to talk about is something that Mallory had said. Is there have been some questions about accessing genetic testing and genetic services, both here in the US and other countries. So I'm going to touch on that. And then we're going to talk a little bit more about like a genetic test report. You saw in our last talk with Dr. Chow, you saw a lot of like numbers, letters, and changes and zinc fingers. And so there is a way to say, here's where my child's exact change is, and here's what it means. So that's what I'm going to try to do, is fill in some of those gaps. And then again, I'll pass the baton to Dr. Wangler, and he'll kind of keep talking a bit more about HAD specifically and the genetics of that of the condition. So first stop is accessing genomic services in the US and Canada. So I'm a genetic counselor. I'm biased. I think genetic counselors are the best, and they're a great resource, especially when you're at the point of having a diagnosis. A lot of my job, or what I'm specifically trained in doing, is taking a genetic diagnosis or a genetic test result and breaking it down to really understand for you guys what it means. It's for a person who doesn't have advanced training in genetics, and I want to teach you what that means. So as genetic counselors all over the country and all over Canada, we have the same body that credentials us, which is why we do things together. And there's a website, findageneticounselor.org. You can go to that website and you can click, I want a telehealth visit or an in-person visit, and it will give you a list of all the genetic counselors you can see in the country. And if you say, well, I live in rural Texas, where there aren't any physical genetic counselors, there's tons that can see you via a telehealth appointment. And just to reiterate, this is to have an appointment with a genetic counselor. We're very helpful, but we are healthcare providers, so you'll need to establish care with us, and then we're happy to meet with you and answer questions. There are also lots of medical geneticists, like Dr. Wangler, for instance, you also can schedule appointments with at various health centers throughout the country. If you don't live in the US, I wish I had a one-stop resource for you. I don't quite, though. There are lots of genetics providers all over the world, though. I know some of them. Many will train potentially in the United States and then go practice in a different country, or there's lots of countries who have their own training programs and credentialing bodies. So you can ask for that referral. This is where Google search does come in, and also this community. You guys have an international panel later. They probably have gotten genetic testing and met with a geneticist, potentially not here in the US. So talk to your friends and use your community. I did speak with some genetic counselors who do practice internationally, and they are currently working on building a repository of where international genetic counselors practice. So they want to basically mimic the Find a Genetic Counselor tool, but for the world. It's in process. Maybe in 1 to 2 years, I'll come back and be like, here it is, it's all done. In the interim, they said, if you have a family who's like, hey, we live here, we're having a lot of trouble, they're happy to try to use their resources to find something for you too. So you can email me. They have my email. It'll be up there at the end. If there's a specific need somewhere else, I can try to see what I can do to help. The other thing to say is there are clinical genetic testing laboratories all over the world also. So even in the US, all the laboratories I mostly use to send testing for my patients, they accept samples from all over the world. And similarly, there's lots of labs in other countries that accept samples from the US or anywhere else. So there is access to genetic testing and genetic services. We can use your community, use us. There are people who can help you find them if you're struggling to find them. So moving on, we're going to do a quick review. Again, we're going to get everyone back on the same page about genetics and high school biology. So just as a quick reminder, we're all made of cells, billions and billions of cells. Inside our cells are our chromosomes, and our chromosomes are basically just our DNA, and our DNA holds our genes. So we have hundreds and thousands of genes on every single one of our chromosomes. Like we just learned, there's 46 chromosomes typically in a human. You get, they're in pairs, so 23 from your mom, 23 from your dad. When you think about the DNA, you think about the genes, it's simply the sequence of the genetic alphabet, which in this case is A, T, G, and C. Those are the 4 letters of the genetic alphabet. And a gene is simply the order of those letters in a specific domain. So you think about your DNA, it's a really, really long strand of letters, like reading a book. It's just that long string of letters. There are parts that are genes, and in between our genes, there's also a lot of letters that we consider to be more regulatory. It's not technically a gene per se, but it's really important information that helps tell our genes what to do. So there's a lot of regulatory regions, and then there's our genes, and our genes are just those letters. And then here's where we like take the next step. Our genes code for our proteins. And when I say proteins, I'm not talking about like your meats that you eat. I'm talking about a molecule. A protein is essentially a molecule, and it's made of these things called amino acids. And every 3 letters of our genes and our genetic code tell what amino acid goes next. So in this like cartoon version, a protein is kind of just like a beaded necklace, and every single bead is a different amino acid. So every 3 letters on our gene, it says this amino acid bead goes next. And then you, if you remember from Dr. Chow's talk, that protein or that beaded necklace folds up in its special way and then goes off in our cells and does its job. It binds to things to find its partner, and it has different domains. So that's like our how we get to the protein. So just to kind of reiterate, this is, Dr. Chow did a great job. She used a neighborhood analogy. I'm going to use a book analogy. We think about our DNA code as a big book. You can read just a single sentence in your book, which is like, for instance, the EBF3 gene is a single sentence or a single paragraph in a book, and you're looking for changes that would change the meaning of that paragraph, or double check the paragraphs even there if you had something like a deletion. You can also do tests, which is reading lots of paragraphs or lots of genes at a time. I imagine many people in this room had potentially whole exome sequencing, which is reading the entire book. It's reading every single gene that we know about in the body, which is about 22,000. And it's looking again for any spelling changes or missing words or things that would alter the kind of the interpretation of that. We now spend quite a bit of time talking about whole genome sequencing. And if you remember, I said the DNA has genes and it has all this regulatory space in between them. That's when you sequence that regulatory space as well as the genes. So you can get a lot more changes because you can, and they're a little bit harder to interpret because we haven't been doing that for as long. And we don't always know exactly how changes in the regulatory region affect a gene. So there's kind of different steps. And I should also say in my analogy, a genome is like reading a book, but also reading every single page number and the footnotes and the copyright, and maybe like the book club questions at the end. It's things that aren't necessarily the genes that you would think about for the story, but it's all the other information and words in that book. So the next thing we kind of think about when we're thinking about genetic testing and genes are gene changes or genetic variants. So whenever you do genetic testing, you're going to see genetic changes in your DNA. We're all a little bit different. It's normal for us to have different changes. Our letters sequence in our DNA is not identical to each other, and that's normal. It's why we're all a little bit different. So whenever you do testing, it's pretty normal that you're going to find changes that are categorized as likely benign or benign. Those are what we say are just normal changes. It's things that make you you, and it's usually things that you don't see on a genetic test report. Things you will see in a genetic test report, though, are things considered pathogenic or likely pathogenic. So for a lot of people or their children, you likely had a genetic test report that said they have a pathogenic or likely pathogenic change in the EBF3 gene. So pathogenic in this sense basically just says this causes that condition or causes this gene to not function as we would expect, and we expect that person to exhibit symptoms. And then, of course, there's always the one in the middle. That's the hardest for anyone who practices in genetics, which is a variant of uncertain significance. I'm not sure if anyone here has a variant of uncertain significance, but essentially it's when we say we don't know. It could be benign, a normal variation that's not affecting the gene, or maybe it is affecting the gene and we're just not sure. We know lots of things about genetics. We also know very few things about genetics, is how I feel sometimes. And so it can be very difficult to interpret genetic changes, especially if it's something we've never seen before, or it's rare, or different types of genetic changes are harder to interpret than others, which we're going to get into in just a minute. So kind of getting into what does this look like? Because as we know, sometimes the type of change and where it is can be important. So what does this look like when you see a genetic test report? So I have two example test reports. They're just examples that I pulled from like major laboratories that they publish freely online. So this is the first example. So the things that you would want to look for on your genetic test report is first the gene. In this case, this gene, instead of being EBF3, it's PP2R, what is that? 2R5D. Just another gene. We have lots, again, we have thousands of genes. So you want to look for the gene. And then I think Dr. Wangler will touch on this briefly, but this is an autosomal dominant condition. And then you look, and this is kind of where I think the numbers and letters come into play. So when you see C dot numbers, so in this case, it's C dot 598G to A. So that means if you look at that gene, in this case, it's this PPP gene, at the 598th letter in that gene, instead of there being a G, it's now an A. So that tells you exactly where in the gene or what number letter has changed and what it's changed from, to from. The thing you see usually right next to it is something that's P dot, and that tells you exactly what protein has changed. So that means when you change a letter, if you remember, every 3 letters codes for a specific protein. So if you change one of those letters, the protein or amino acid code may change. So now your beaded necklace, it's going to fold up. At least one of those beads is now different than what we expected it to be. So that tells you what number and which amino acids changed. So those are going to also always be on your report. And if you're looking for things about like, well, is my child has the same, change as someone else, this is what we'd be looking for. So you have the exact same position, is the letter change the same, and is the protein change the same? The other thing then that we just talked about, in this case, usually you see the interpretation, pathogenic, meaning causing that condition. And then the other stuff on here that I put in red really is more for me than it is for you. So this kind of tells us how the lab made the decision to call this change to be pathogenic or likely pathogenic. This is really important to us as a clinician, especially if there's a variant of uncertain significance. It's really helpful information for us to interpret what we think it means. So we're going to show you just another example, just because we're learning. This is actually a test report from a laboratory based in Germany that I've seen lots of reports from in my time. So again, we can get genetic testing all over the world, and it's typically very similar. So again, the gene, in this case, the gene is LDLR. And then here you see the variant coordinates. You can see the C dot. So at position 1,135, that letter, instead of there being a T, it's now a C. And when you change that T to a C, you can see the protein has changed. So at the amino acid 379, that has changed to a different amino acid. And you can see, we didn't really touch on this one, but we can see it's heterozygous. Heterozygous meaning only one copy of that gene is affected. You see the word homozygous, it means both copies are affected. EBF3, we're going to expect these to be heterozygous. And you can see it's a pathogenic change again. And then here's all the other interpretation information that the laboratory used and I would review to basically say, how did we figure out this was indeed a pathogenic change? So the other thing I kind of want to touch on, which I think is, again, we're learning, we're more advanced, are different types of genetic changes. Because when we start thinking about the conversation that we were kind of having before about therapeutics, interventions, we got to understand what the change is doing to the gene or the protein and how it's affecting it to then understand how that could change its function. So these are the, I think maybe the only types of variants that have been found in HADS and EBF3. Last I checked, it was the only ones that have been found. So they're just different types of variants. And so we'll just kind of go through, if you have a missense variant, that's the kind of on the reports I just showed you. That's when one letter in that DNA code has changed or switched. So if you believe like our original gene that we would expect, it says the cat had red fur and ran far. Well, now let's say that C changed to a Y. So now it says the yat had red fur and ran far. I don't know what a yat is. So that has changed the function or the meaning of this gene. And likely we could potentially in our world interpret this as pathogenic maybe. There's also something called nonsense. And so sometimes those 3-letter codes, there's always going to be 3-letter codes in our DNA that tell us to stop. That's the end of the gene, or that's the end of how long this bracelet of, amino acid string needs to be. They're all going to be different sizes. So sometimes we have to figure out how to make it stop. And the stop is a 3-letter code. And so if you make a genetic change and you introduce that 3-letter code too soon, well, then you're only going to get half of the gene, or maybe even less. So in this case, we would assume that there'd be a change at approximately the F position in fur that introduced that early stop. So now we don't have, that protein essentially is too small, right? Like we didn't transcribe enough amino acids. A frame shift is when you usually have a deletion or duplication. So you're adding in letters or you're taking away letters. And so that changes your 3-letter phrases after you make that change. So in this example, we took the D out. Well, now every single 3-letter code after that has changed. So now we would think that those amino acids or those beads would also be changed, and that can alter the function of that gene. And the last one is intronic. And so if you remember, this is kind of a change more in that regulatory region. So our sentence or our gene was still fine, but right outside of that gene in kind of the regulatory region, in this case, it's our additional sentence in parentheses, has a genetic change. And we do know that that genetic change can affect how the gene functions. And again, this is important because it's when you're thinking about how do we resolve this, or are there interventions or therapies for it, we're thinking about different things. So if you have something like a missense change, well, we know the protein, that bead, we expect that whole beaded protein necklace to be folded up as we'd expect, but maybe it can't do its job. Like it's maybe in the anchor region and it can't bind right. So we still have the product there. We just need to help it function better potentially, which is different from, let's say, a nonsense change where maybe you only transcribed a quarter of that protein. You basically don't have enough, or it's completely gone and missing. So an intervention for something that you just have too little of is different from an intervention where something exists, but maybe isn't doing its job, all of its jobs as we would expect it to. So we also kind of talked about how knowing, again, what type of change you have and where it is can be important too when you think about genetics. So this was kind of just a really quick overview of how we interpret genetic test results and what they can look like and the different types of variants. There's still a lot of research to better understand exactly, again, where your change is, what that outcome means, or if the type of change is really important for how that person or how that gene functions. So I'm going to go ahead and pass it off to Dr. Wangler, who's going to go through some more information about genetics, and then we will take questions together at the end.
Dr. Wangler completed his medical degree at Baylor College of Medicine in 2006 and his pediatric residency at Texas Children's Hospital in 2009. He is board certified in both pediatrics and medical genetics. He is an associate professor and physician scientist at the Jan and Dan Duncan Neurological Research Institute in the Department of Molecular Human Genetics at Baylor College of Medicine. His research interests include rare and undiagnosed disease and using genomics in medicine. He is an expert in HAD syndrome as well as other parotomal disorders and genetic brain disorders. Please welcome Dr. Wangler.
Okay. All right. Thank you all. Thanks to all the families who are involved in this organization. And it's great to be back. Haley and I have sort of given a similar session in the past, and things have really advanced. I'm going to go over some of the things, and maybe, you'll see some of the things that you've already heard again. Just going to try to make it a little more maybe relatable for HADS and your specific situation in the Genetics 101. So I was going to try to do 3 things. First, kind of talk to you about how can you understand your specific DNA results. We'll do a little bit of terminology and a little bit of breakdown of a HADS result, and then I'll go back over. Dr. Chow showed you the large kind of clinical paper that was done here, and I'm going to go back over some specifics there and talk about what we term genotype phenotype correlation. And then I'll try to address some of the questions about like therapeutic horizons in very general terms. I'm hoping to give you like some ideas to kind of help you navigate when you see things in the newspaper, for example. So the first thing I wanted to mention is that since, you know, we've really over the years, it's grown into there's HADS, but also sort of the 10q26 deletion. So we may have families in the room who are diagnosed sort of with different genetic tests. The two major tests that I think we're seeing in the clinic that, you know, sort of flag you for us are first the chromosomal microarray. So the chromosomal microarray is designed to detect deletions, duplications, and rearrangements. So up here is shown a diagram of like one of our chromosomes. This would be chromosome 10 in this case. And what this is showing a deletion. This is when a segment of that chromosome has been essentially removed. This actually is a infographic showing a tiny deletion, actually a single base pair deletion. That would be, you know, different, but you can see that deletions could range from one single base pair all the way up to, you know, a large segment of a chromosome arm. And then so some families may learn about their diagnosis from a microarray that detects, like, for example, a deletion. And then other families may learn about their diagnosis from whole exome, which Haley covered, which is essentially going to be very good at detecting point mutations. So what's shown here is the type of mutation that an exome may uncover in a gene. So the one thing I would like to emphasize is that, you know, we showed you these DNA reports. I think you should obtain your DNA report. We are not going to administer a quiz to you on your report, but I think having that record is very important. First of all, medicine changes, technology changes, maybe your doctors change. The DNA report is going to contain information on the code that's not going to change. Even if the nomenclature changes, you know, it can be figured out. So it's a very solid kind of document that you have. And, you know, if things change, you can always sort of share that document with the healthcare provider and, you know, with sufficient sort of investigation, that should lead them to the latest information on, you know, your loved one, your child. So I think it's important to actually take command, have your DNA report. This is an example, maybe one of the first ones that we saw. So there's some terms that I'll just go over. So heterozygous. So in this condition, the patient has sort of what we'd think of as like a clean copy of the EBF3 gene. It has no mutations on it. And then there's a second copy that has a mutation. And so heterozygous means like the two are different. There's one sort of clean copy and there's one copy with the mutation. And then pathogenic, Haley went over, there's criteria that the lab will use to judge whether a mutation is pathogenic, whether it's a variant of uncertain significance. And these are things that's why we have jobs in genetics and genetic counseling, is sort of looking at the evidence for those things. And then also de novo. So we'll go over that, but most of the reports are going to call out whether the variant is de novo. De novo would mean that they identified the change in the patient, but then the parents did not have that change. We'll go over a little bit more about that. Oh, other terms you might see, for example, novel. So novel means like your mutation, we haven't seen it before. So it could still actually be called pathogenic or benign based on other information, but novel would be we really don't know much about it. All right, so this is sort of leading us into what we call, like, can we establish genotype phenotype correlation for HADS? So and the reason that that's important is when you get that test report and you look at it and you sort of see the change, you know, it may lead you to, okay, it's HADS or it's 10Q, but then you're sort of like, well, what, you know, can it tell me anything else? Is it severe? Is it mild? Is it moderate? What do we know about the mutation? Is there any predictive ability of knowing my mutation? And so we call that genotype phenotype correlation. Phenotype is the outward characteristics. So I'll go over a few of the phenotypic features, and then genotype, that's the exact change or the mutation. So so this is what for chromosome 10 deletion, this is kind of showing the chromosome arms. This is chromosome 10, the long arm, and the Q26. And one of the determinants of like genotype, phenotype correlation could be how many genes or how big of a segment of chromosome 10Q are missing. In terms of inheritance, these conditions are dominant. And so again, dominant means that in the heterozygote where there's one copy that has mutation, the patient has the condition. We call it autosomal dominant because it's on an autosome, so it affects both boys and girls equally, and individuals with the mutation will have the trait. And, you know, we have seen examples of both inherited situations where there's an individual with the condition and they pass on the affected gene, and that's with a 50-50 chance. And then more commonly, we would see de novo, which is basically one individual has the mutation. The parents don't appear to have the mutation. What about, I'm going to get to, I'm going to talk a little bit more about mosaicism in a second. So what caused so for a lot of families, they wonder like, well, what caused the de novo point mutation? And yeah, you can read about that there's like radiation and X-rays and UV rays can mutate DNA, and there's chemicals and what's going on with our food, what's in our food. But by far the biggest factor is DNA replication. So it's just something that's like part of our genome. So basically our genome has to copy itself over and over again. There's 3 billion base pairs. For example, the average sperm cell, the genome like 50 times in a teenager. For a 40-year-old male, there'd be thousands of times that the genome had to copy itself. And so we know that, you know, everyone has about 3 de novo like coding mutations because of that. And it just matters sort of where did that occur. So for so epidemiologically, we can say that like advanced maternal age would increase the risk of chromosomal abnormalities. So that would apply to sort of 10Q, and then advanced paternal age would increase the risk of those point mutations. It's important though, we always try to discuss this in clinic that, you know, neither parent really controls any of that. Neither parent controls which gene they have passed. And we do think that a lot of times families will think back to certain events early on, and we really think those kind of diet and exposure issues have pretty limited impact. It's sort of nothing that you did or didn't do that caused this. It's sort of just part of how the genome works. I will just cover mosaicism if it comes up for people on their reports. So germline mosaicism, that's where there is a parent who is at risk for having more than one affected child, despite the report maybe saying that it's de novo. So remember, often the parent was tested from a blood draw, or maybe the parent did a buccal swab, not able to test every cell in everyone's body, right? We don't have that kind of knowledge. So there is a risk that some cells could have the mutation, and you could have an individual who, appears negative in blood, but has some cells with the mutation. This is pretty unusual. In most genetic disorders, we would say like less than 1%, but it is something to consider when we give you like the counseling about de novo, because a lot of times people would think, well, it's de novo, so that means it, won't happen again. And in general, that's true, but sort of with 1%, we give that sort of caution. And then somatic mosaicism would be like if there's an individual and we can detect that there's a proportion of their cells with the mutation. So, they sort of maybe have attenuated symptoms in that case. Important to remember, though, that like a person with mosaicism, you only get to pass on one gene to the next generation. So you don't pass on some mixture. So mosaicism doesn't like continue down generations. So Dr. Chow went over in a lot of detail, like sort of so if we go back to the phenotype, how is HADS described in the medical literature? So, the names, the hypotonia, ataxia, delayed development, and those are important features, but there's also, other very clear features. There are, we would say, dysmorphic facial features. There's a pattern of facial features that we can detect. Some of the patients have kind of deeply set eyes. They can have sort of low set ears. There's GI symptoms. Dr. Stanisel will talk about genitourinary abnormalities, or I think she will. And then global developmental delay, ataxia, and then we talked about like the muscle tone and speech delay. So these are kind of some features that are, if you sort of reading the literature about the condition, you get your test report, you Google, you find something, a lot of these things are going to be described. But, I think to emphasize the importance of the research is that we really need to continue to document the clinical history in more detail. The more we see the patients, the more we can see variations. First of all, there's things that are very consistent in the pattern of the condition, but also we learn new things all the time. Like, sort of we don't really know how to deal with, competitive swimming, for example, here today. Also, it's very important that the research continue because I'm going to go into sort of the therapeutic horizons, but we really need to know very well the full range of this disorder, because if you think about it, any therapy that would come down through any technology, we're going to have to study it scientifically. We need to know, does it work? And in order to know whether it works, we sort of just came upon this condition, in sort of 2017. So we really need good data on what is the condition like without the treatment? What is the condition like with the treatment? So that's very important. And that's sort of why you see so much energy being focused on that. And so I would just plug again that you reach out to Dr. Chow's team about the study. So this is just showing like some of the data more data from the genotype phenotype study. It shows, sort of the gender distribution, the distribution of age at the time of diagnosis. We may see this change over time as a lot of patients will probably be picked up earlier. You can see that this affects multiple ethnicities, and we would expect for particularly de novo mutations for those to be pretty randomly distributed across all ethnicities. You can see that there's, well-defined like patterns that emerge for growth. And then, this shows you the exact distribution of where, like, say, your test report falls with, how many are de novo, how many have, affected parents. And then these, don't worry too much about these, but there are ways that we can rate mutations in terms of how pathogenic they are. I also thought this was really nice, the way that we're able to sort of see how, what percent of individuals had these key features that I mentioned. So this really helps us define kind of the condition in terms of like how likely we are to expect a patient to have these sort of phenotypic features, gastrointestinal findings, eye findings, renal or genitourinary, and then the muscular or musculoskeletal findings. So all this led to the graphic that Dr. Chow showed you earlier, where we can actually see some signal, which is very interesting, of the location within the EBF3 gene. We can see a location where it makes a certain component of the protein, and it seems like mutations in that area have some, higher severity scores. So this kind of data is like very important for us to establish early on, because again, down the road, we sort of want to know, can we give families more detailed predictions? And then also, as we start doing treatments, this seems to say that we'll need to sort of collect genotypes for anyone that is being treated. So I'll end with kind of what are the therapeutic horizons. And this is, so this slide probably looks sort of technical, but to me, it's actually meant to simplify, because what happens is that you go to the newspaper now, and or I don't know if anyone even reads the news. Anyway, you pick up your phone and open the app to whatever news source. And there's a lot of stories now about gene therapy, gene editing, oligonucleotide therapy. And I think it can seem confusing. So I'm a geneticist, so I just organize it like, okay, are we doing the genome or the RNA or the protein or the disease? So that's kind of how I think about it. So if you start off and think about that, the patient has a mutation in EBF3 or a 10q deletion, that's the genome level issue. And yeah, you can read I just saw an article today about CRISPR gene editing launch trial for a condition called progeria, gene therapy, where we're going to deliver back kind of the missing gene. And then if you think about that mutation leads to maybe reduced RNA or mutant RNA. And then you can read about that there's therapies that are being developed that would target sort of abnormal RNA, or maybe even enhance the expression of RNA. If, in some cases for 10q deletion, you would think we would just have reduced EBF3 protein. Maybe some of the mutations, there's an altered protein. And so you could think about, are there small molecules or chaperones that could help that protein function better? And then there's kind of all the other, things, which is collecting data on the patients and figuring out how can we help the patients. And that I would think of as sort of symptomatic therapy. And I was just going to go through each of these and to give you a sense, because I think none of us know what will happen. We don't know sort of the tech, we didn't know that CRISPR would come along, sort of 15 years ago, for instance. That's not something we could have predicted would come along. So we don't know like when some of these things would become more feasible or available, but we can think about what are like the problems and what are the like advantages. So if we think about kind of, if we're going to try to fix the gene, that'd be great. I mean, there's a lot of advantages, right? We would correct the root cause. We could restore the EBF3 in the cells that we could treat. That would maybe last for the cell's lifetime, depending on how we delivered the edit or the gene therapy. So it sounds great, right? If we are going to try to silence a mutation, Maybe we could use an oligo, and that could maybe shut down a specific mutation. If we think about chaperones or small molecules, these would be nice because we can give them by different routes. We could give them orally. We could use drugs that are already available. And then symptomatic therapy, I mean, we should be implementing that and improving that today. So that's has a strong advantage that we already know sort of that we need to do that. And we already have the tools to do that. But if you think about like the challenges, I think you can see that like sort of the further up you go, the more challenges there are. Whereas the further down we go, the more we can probably do today right now. So for genome level therapy, I mean, a lot of this is not developed or approved yet. There's certain companies working on certain specific disorders. When those disorders, you know, see results, maybe that should feed back into many genetic disorders. But we still would face that we don't have a great safe way to deliver to cells like in the brain, particularly deliver to all of the brain. That's going to confer a lot of risk. You're talking about things like putting a virus into the brain cells and stuff like that. And then also a key question that Dr. Chow brought up is, are we delivering it at the right time? If we have a patient in clinic, is it that we needed to deliver that gene, you know, several years ago? For the oligo, again, those are not really developed or approved yet, although, you know, there's certainly diseases where that is being used. But again, we'd have some of the same questions about like, are we delivering it at the right time? And we're going to have to do repeated dosing for those. So we really have to think about delivery there. For the small molecules or drugs, you would say, okay, well, this may not address the problem because we're not fixing the mutation, but we could give repeated doses. And then for symptomatic therapy, you know, it's not really addressing the cause, but it's just going based on the information that we have. But this is likely like one of the more important things we can do is focus in on specific clinical problems and try to figure out how to intervene. So I think that's how I hope that simplifies and doesn't complicate it for you. I think that's all I wanted to say, other than just, you know, your participation in the research is helping to develop like very detailed natural history. So whatever of these sort of routes starts to become available, the data that we're getting now is very valuable. And then also I think support for Dr. Chow's like rare disease, you know, sort of animal models for this are also key, because if you think about that question of timing, when do we have to restore the gene? Those are things that we definitely can look at in animal models. So I think with that, I'm ready to take questions. I think Haley is here as well.
Hi, I'm Joanna, in case I haven't met anyone yet. My question is that we were diagnosed in 2017. Is there any benefit to rerunning the test if it's banked, or getting another exome because of technology and how it has increased? What would be your recommendation of an old test versus getting a new one?
Oh, that's a good question. I could just tell you that I think generally if the diagnosis works, and we're comfortable, like, you know, this is the diagnosis. We were doing pretty good sequencing, you know, by 2015. So we probably wouldn't rerun. Where we would rerun it is when a patient shows up with maybe a new finding or something doesn't fit. So for instance, maybe there's a new manifestation. We don't understand this manifestation, and we're looking for, maybe there's a second gene that we didn't know about back then that we would be able to find now because of better knowledge. Also, I guess if, it's basically going to be a matter of if the clinical picture for the patient kind of, fits, or if there's something else that we want to investigate. But so I guess to give a more simpler answer, I think the technology was pretty good probably for everyone who was diagnosed. A lot of the advancement has come in like learning about new genes and new knowledge and new kind of computer algorithms. But if there is something really unusual, we might sometimes pursue like reanalysis, new testing, that kind
You
of
need to
thing.
go.
Okay.
Hi,
Was there
my name is Rich Holmes. If I can ask
Yeah.
you a question.
Yeah.
All of your proposed interventions except the first one to change the mutation wouldn't be effective unless you already knew the condition existed.
Yes.
So that isn't typically something we find out in utero. So it's only after birth. Do the studies in the animal models, which I assume is the only place we really have conducted them, show that the defect in EBF3 continues to do harm after birth, as opposed to the structural damage caused in utero, which is in place already?
Yeah, I think you're asking the key question that is what I'm trying to say is that's the reason why it's so important to have the model, because I can think of examples for other genes where, there's sort of maybe, let's think about the brain, and the lack of the gene or the mutation in the gene leads to an abnormal development. But then there's also from there kind of maybe a degeneration where there's a loss of neurons. And you can think of situations where, well, I can't fix the developmental issues from the past, but I can prevent the neurons from sort of degrading or dying with this. So we're not seeing that type of example for this condition, but that's why it's very important to have work on the animal model, because we can't really answer that with human studies. I guess I would say also for, it's likely that we wouldn't know in utero, as you said, because particularly in situations where there's a de novo, so parents could have sort of carrier screening and prenatal screening, and it could still develop. But we probably are going to be picking up patients like earlier and earlier across time. So we'll have to see if that, gives us windows of opportunity.
Thank you, Dr. Wangler. Thanks for your presentation. I'm curious, there's that promise that right now seems so distant of gene therapy. Are there examples of other types of gene therapies that are currently being developed that are maybe ahead of where HAD's research is, that maybe we can look at how it's being used in other areas?
Yeah, I mean, I think I wouldn't present these if it was all just kind of, going to another planet sort of level, because there are, for instance, like spinal muscular atrophy, there is a gene therapy that's, currently in use. There's been trials for disorder like giant axonal neuropathy. So I think some of the things that are learned in these conditions are that like there's benefit, like for spinal muscular atrophy. I mean, the neurologist probably knows much better, but there's benefit, but it's not like a complete correction by any means. It's sort of like you sort of reduce the severity for the patient. So, I think that to me emphasizes the need for really good natural history for a new a newly described disorder because for spinal muscular atrophy, there's a lot more data before to classify the patients by severity. When the therapy rolled out, it's much easier to sort of see the benefit. If we don't have good natural history, you know, it could be you don't have the ability to really say whether it works or not, you know, particularly for something that works partially but doesn't take away all the consequences. But yeah, these technologies are you know, they're here, and I think for us, it's like we're not necessarily working on like viral vectors to the brain type of research, but the research we're doing is sort of maybe establishing, you know, when to treat, how to treat, and those are going to be the important questions, because my sense is that a lot of biotechnology development will push some of these treatments out for many disorders in the coming years. Yeah.
Can I ask just a quick question about, so with EBF3 being de novo initially, what about so kids then who have EBF3, if they decide to have children down the road, is there an increased possibility then that this becomes something that they pass on, even though they inherited it de novo?
I should let you answer some questions. I'm just talking. Okay. So yeah, this is great. Because once the patient sort of has the de novo mutation, it's, you know, assuming they're not mosaic, they have it in all their cells. So they actually move into a category where it's a 50-50 chance with each pregnancy of passing on that mutation. Even though they're sort of the first in the family with this mutation, they have it in all their cells. So they now have a 50-50 chance of passing it on.
And we got a question from the from Zoom on the chat there. They say, can you talk about how and when something like CRISPR would be used?
Yeah, I mean, so no, I have no idea. If you think I know what's going to happen, I have no clue what's going to happen. That there's just, you know, I was just reading this article this morning about a trial for progeria using CRISPR. So we call that gene editing therapy. So there you're delivering, like, you could think of it as like you're essentially delivering like an editor to change the mutation rather than, you know, delivering back the whole gene, for instance. I think there's a big push to do this for several genetic disorders right now. But yeah, I don't know, you know, how applicable it will be to HADS I don't know yet.